for the organizer to invite me for this. So it's going to be a pure methodological presentation, and I'm really happy that I'm coming straight after Mark, because he has, it's the best introduction I could get on this part. So what we are looking at CIF is we're trying to understand how the immune response operates, and for this we have to understand the cellular immunity. And from the beginning on, then we have a big issue, is that it's highly complex and you have lots of interactive partners beside what Mark said about plasticity. So why that? Because actually the immune response is getting driven by multiple hematopoietic lineages that are themselves subdivided in multiple subsets within one given lineage with different functions. And each of these different subsets, they might have multiple stages of activation. And they even have maybe a different metabolic stage. And then now you have to put the environmental cues into perspective, and that could also change the way your cell is responding. So you have to find one way to actually tackle this high complexity and study your cell. So that's the different ways to enrich target cell population here. So you have very classical ones like panning and pre-plating. That's what people sometimes do for macrophages. Also erythroylation, which is a kind of mix of centrifugation, and based on the cell size and density of the cell, you can sort cells. Laser capture microdissection, microfluidics, which is really emerging, magnetic bead selection with the drawbacks you have heard today, and fluorescence activated cell sorting. So now we'll only focus on this. So why is MFC, so multi-parameter flow cytometry, such a great tool for microgenomics, at least in my point of view? It's because you have a lot of available markers, so that's really a big strength, at least in mouse and human. You really have lots of choice to assert, to test to sample yourself. You have a lot of dyes and probes available to check for viability and apoptosis markers, and many also metabolic function you can assay with probes to test that, and you can look at that by flow cytometry. Now you can do more than 9, you can do up to 20 parameters simultaneously. Actually, we do 17, but a new level actually has been reached, because there was we'll be at CITO meeting where I go after that. Someone said that they could do 27 by flow cytometry. So we are just pushing the limit more and more. So that we are coming into a possibility to do cytomic studies. So like what you guys do, but now on cellular level based on cell surface molecules. And we are able with this also to trace minor populations, and also combine this with lineage tracing studies. At least in mouse, we can tag specifically genes with fluorescent reporters, and combine this with the detection with cell surface markers to actually precise or study more the function of a cell. And of course, you have FACS cell sorting. So that's how it could look like. So you come out with a very complicated gating strategy, and you, Mark, illustrated that it's really critical to put as much marker as possible to actually be sure of the purity of your cell. And of course, the aim, and that's why you guys are all very familiar with, is to actually do transcriptomic studies to infer correlation of gene expression, to have a clues on what are the molecular mechanism and developmental choice, or that's help you to understand the cell interactome. Well, what I will try to show you is that if you combine actually high content flow cytometry, then you have an unmatched phenotypic precision of cells, and you combine that with FACS cell sorting, then you reach an unprecedented purity of your cell population. So I will first now talk about multi-parameter flow cytometry. One of the main take-home message I would like to give you today is that you should consider this flow cytometry not only at this part here. It's really the whole part. It's the whole full experimental design that is really crucial to ensure good quality data. Because as Mark mentioned, cell preparation, depending on the type of enzyme and things you will take to isolate your cells, you might cleave some markers, you can increase your the contamination of your cell subset. Then also you can alter very badly the viability of your cells, and of course this is not good for the kind of study you guys want to do. And then finally, if you stress also, if your procedure is very stressful or very lengthy, for sure this will have impact on the genetic profiling you will look at by transcriptomic. So really, flow cytometry is all this. It's the cell preparation. You have to standardize this very much. The cell staining. You have to test what you do on the flow cytometer first to ensure that what you do is right. And then you have to go on the cell sorter, sort your cells very, very quickly and extract DNA and RNA. So just to show you that what I'm saying is actually not, one of the things also is that by adding a lot more markers than what people used to do in conventional flow cytometry, I'm not talking about 6 or 8, I'm talking about 12, 15 or even more, you actually bring a lot of robustness to your data set, because it's kind of correlation like in a geographic map. You have many, many coordinates that makes that at the end, if you sort the cell that fulfill all the criteria you are giving, then you increase the chance that you have the right cells. And one of the things is that you have variability from instrument, and this we have quantified. It's around 3% of your measurements of flow cytometry. And what is here, this whole part, we believe it accounts for 30% of the variation. So really, this work on your protocols to extract cells. So just to show you that this is actually working, so here it's a 17-parameter flow cytometry panel we have developed at CIF. In one go, we can determine 26 population, and it's with 15 colors only. And you see all the population we can look at. So why I put this example here is because we applied a very robust extraction of cells. It's standardized. We use machines to do that so that we don't take the operator variation into the process. And by doing so, we were able to, for one of our customers, because we do service of immuno-phenotyping, we were able to pinpoint one mutation, which is the little thing here. This guy came to us and said, you know, I look in the thymus, the thymus is hypocellular, so for sure there is a phenotype, but I have no clue what's going on. If I do my FACS studies, my ratios are similar. And we actually confirmed that. You see, when you have the blue is the control mice, and the orange is mutant mice, and where the two lights are overlaid, that means you have actually no difference in terms of frequency of the cell subset that is being assayed. Now you see that this little blue point here is actually different, and when you look at the FACS profile, that's how it looks. So you have like 250 cells here, and here in average is 25 cells. Just for you, this is the most immature primitive precursor that migrates from the bone marrow into the thymus, and it accounts for less than 1% of the population. And because we were so robust in the experimental design, we are able to make a call on the phenotype on 25 cells out of 1 million, in comparison to 250 cells out of 1 million. So take-home message here is that you should, by increasing the number of identifying markers, you improve separation, and you increase also the precision of your phenotyping. And you also decrease the chance to contaminate your population, because basically you would put markers that before you would not be able to put because you have technical limitations. Now you can put it to actually gate out the cells that could contaminate your samples. So what are the critical steps in multi-parameter flow cytometry? One of the things we as researchers most of the time completely undermine is that we don't know really well the instrument. And I think that's a big, big issue. Because first of the things you have to know is what laser are equipped on the machine, what kind of fluorochromes you can detect, because from one machine to another, it's virtually every machine is different. So you really have to understand that. Then you have to characterize your instrument. We see lots of mistakes because people actually don't work in the valid range of measurements. So for each individual PMT, so photomultipliers, you have one zone which is dark zone, which is the electron where you have the electronic noise, so the measurement will not be accurate. And you have another one which leads to very drastic errors when you set up compensation matrix. That's the maximum of linearity of the PMT, and you should also not be in that range. And most of the time, people don't know how to look that. Actually, most of the platforms that do flow cytometry, they quality control their instrument every day. So this is, you just have to go to them and ask for the data. It's there. Then you have to know the instrument sensitivity, because you have here we have that's all the parameters we have on our Fortesa. So you have the Bibles from the commercial companies that say, oh, this fluorochrome is more intense than another one. But now I will rely on your instrument. Well, it is here. Actually, one of the most sensitive dye is BV421 currently on the market, and you see that on our own machine, it's performed really badly. So you have really to take this into account to actually maximize the separation you get afterwards. And maximizing separation is crucial to ensure a good purification of your cell. As I said, you have whole arrays of dyes now that are available, so it makes the whole thing a bit complicated because you have to go into panel design. We'll touch down a few words on that. And some people actually do that, and not many people actually do it because it's costly and time-consuming. But you test many combination of fluorochromes and on cell surface molecule to actually minimize effects due to fluorochromes that are spread data. Very crucial thing that also lots of labs don't do, and they should do, is titrating reagents, because if you have a reagent that you use at this level or at this level, of course you will contaminate your negative fraction with positive cells of one given lineage that you maybe would like to exclude because you put this marker in your panel. So titration is crucial. Then resolution of staining is also important. What matters for you is the separation between positive and negative. That's fairly easy, but it's not only the thing you should take into account, it's also how your negative cells spread, because if you have an intermediate level that is somewhere here, and you have a big spread of your negative, big standard deviation of your negative cells, then you will contaminate your intermediate populations with negative cells. And then you have to run appropriate controls. So in each experiment, you should have bit control capturing the antibody you use to label your cells. Very crucial now with high content multi-parameter flow cytometry, the gating control, the FMOs. I will give an example on that. And then if you do stimulation, because cells could change their morphology, they could change autofluorescence, you have to have biological controls, unstimulated, stimulated, and that is lacking in many times. And then another tool you can consider, especially for you guys that do maybe cell sorting to purify cells for microarray studies, you can actually, each time you do an experiment on a cell sorter, the sensitivity of the instrument will vary. So you have to limit the variation of the instrument because of all the implication on your panel. And you can standardize the instrument now. There are tools to do that automatically. So just that you are aware that it exists, and it's very powerful. And then, of course, we come to cell sorting. So what's the main issue is compensation. What is compensation? So very simple view here. You have two fluorochromes that are close in the light spectrum, the FITs and PE, and you see this zone here of signal coming from FITs into the PE channel. Well, on the PNTs, we detect just a part of the wavelength on one PMT. So we need necessarily this will be excited as PE at the same time on the cell, and it will give signal, and it will pile up in these parameters. So now we need to readdress signal into the correct parameters, and we do that by calculation, by subtracting the values of one parameter from another one. What it helps to do is this kind of thing. For example, here is one dye which is CD19 FITs, so staining without CD45 amcyan. And if you put amcyan, what you see now is that a negative population becomes intermediate. And it's only due to this compensation issue. So, and just one important thing is that to limit this, fluorochrome choice is key. And you have to compensate. So how it works, so here is just an example. You look at several parameters versus the one positive for your cells. You compensate, so actually this cell look double positive, but upon calculation, now you can bring it back to where it is. But I put this example in purpose because you see now we have another issue, is that the cells, they spread a lot. So we have from 10 to the -3 to 10 to the power of 3. So it's tremendous range where your cells are spreading. And that's actually bring errors in measurement or inaccuracy. So it does not serve the spread data, and the bad thing is that all this compensation into the multidimensional space is actually cumulative. So that means if it's really bad from the beginning on and you put on markers, the resolution of your panel is going to be really crap. So you have to improve that in a way. One way to look at that is to use FMO controls, as I mentioned before. So FMOs are meant for fluorescence -1 controls. So basically you put all the antibodies that are in your mix except one, and we do that most of the time for continuums or markers that are not well defined, or when you have an intermediate population. And what you see here is that what is said as being negative here with the FMO control, now you shift your negative cells a little bit, and you don't want to sort these guys out because they are negative. When you put your positive marker now, your positive cells are here. So they are really crucial in cell sorting strategy. Then the other thing is that you have to have in mind the scheme you want to sort, and using this you have to find the best combination of fluorochrome possible. So here is one example. It's just the immuno-phenotyping splenocytes, and we want to look at all the cell subset. Now, if we look at all the different fluorochromes, we actually minimize what I told you before, the compensation issues and the spread data error that is associated with it. And another strategy would be, in an ideal world, if you would have a machine with 25 lasers exciting on each individual one fluorochrome, you could have one fluorochrome per laser, and then you have no issue at all on compensation, and you see that even very dim population expressing like the NKT cells, CD161, and dim for CD5, we are able to fish them out, and that's a 13-color multi-parametric panel. So that's possible. Now about checking the sensitivity and fixing it in time. So two ways. One way here is a traditional way using beads. So you use bead particle actually they emit strong fluorescence in all the channel of your machine, and you can you pass them without compensation, and you have several beads with different fluorescence intensity. And then what you say is that you set interval gates and use this as a scaffold for your standardization. So you will monitor the mean channel value of this peak on day 1, and then on day 5 you will pass exactly the same beads, and you will just make sure that your beads they fall in the exact same channel, and that may tell you that you need to push PMTs, the voltages and stuff. But what matters at the end is where your positive population falls, because beads, they don't change in their properties, it's physics, they are intrinsically fluorescent. And like this, you can completely standardize the instrument variability, and that's a big help, especially when you do cell sorting and you want to compare things. You don't want to compare the variation associated to the machine, right? So now set up the experiment, gating strategy, you have to validate it, so that's a nice thing. So on BD machine, you can use application settings. If you standardize, as I said, on BD machine, it's a lot more well thought, so it's already included. Then you can compare results over time, and just again to depict that I'm not saying crap, this is actually known for decades when people doing clinical work. Here we run exactly the same panel in June 2013, September, and January, and you see that, okay, we might have some variation, but that's due to the operator that makes pipetting errors and not due to the measurement of the machine. You see that the negative cells and positive cells, they always fall in the same place. So this is really powerful, and you should really aim to do that. Now, that's multi-parameter flow cytometry, and we want to combine it with cell sorting, right? So what's the aim of cell sorting? The aim are to sort all cells of interest in the sample. So that's, of course, in an ideal world. You want to have everything, because it's going to be the very, very tiny population, and you want to study it. Then you want to only sort this, and that's all, again, an issue that we always have to face. And you have to sort fast also to ensure high viability of your cell if you want to do functional assay, or as what the question we had before, to minimize the effect due to the lengthy procedure of extraction. So what we say by doing this is that we have to work on recovery, we have to work on purity, and we have to work on speed. And the bad news is that each of these individual parameters, you cannot have them all when you do cell sorting. So you have always to make compromise depending on what you want to do. So how is cell sorting? Well, basically you stain your cells. All this maybe you know already, so I will be very fast. They come into a flow cell where they are hit by lasers, and then they are moving into, that's electrostatic droplet cell sorting. You have other technologies, but I'm talking about this one. So then your cell is, during that time, in a fly zone in a stream of liquid, and then you will have drops that will start to be done. And at this drop break-off, what happens is that the machine and has analyzed the cell characteristic by fluorescence, and it will charge droplet with electrostatic charges that allows you to deflect these droplets into your tube of interest and then collect your cell. Well, one of the things is that this does not happen very steady, always at the same time. You have variation in this drop delay, and actually when you look at that time point here, you see that you have a Poisson distribution. And that's actually really important because that will, we will need to talk about sorting mask for that. So what are the key factors to influence the cell sorting quality? Well, you have a whole instrument set of parts, which normally are taken care by the flow cytometry platform when you are going to, and then a sample preparation. So again, I stress that you should have top quality samples. So one of the main things is because we do single cell analysis, so it has to be on single cell stream of cells. So if you come and you have packets of cells that are clugged to one another, it's not going to work. You have to avoid high frequency of dead cells because this is also already detrimenting yield and recovery and also purity. And you have to match the cell concentration to your instrument and set up a cell type. So I will give some clues on that. Now, if we take our criteria we had before, what influence what? So the nozzle choice, for example, nozzle is this part. So that's where the cells pass through. That's a hole basically that allows you to have a stream of liquid. This nozzle choice is critical for speed because if you have it very, very tiny, then you can put on a big pressure and push cell very fast through the stream. And if you need to change the nozzle size because your cells do not support this treatment, then you will sort at a faster speed. Then drop delay, because drop delay, if it's not well set, you will have purity issues. You will sort apples and pears and put them together in one tube. So this one we don't want. And then one of our paraglider net is the sort mask, where you actually have to choose if you want to do recovery purity or if you want to maximize P. And what are sorting masks are basically the electronic decision that the operator has to make for you. And you have 3 modes on which you can run a cell sorter. One is a yield mode. Basically what it does is that when you see an event here that comes at this critical point of the drop, when the drop has to be charged, and if it sees that you have a cell into that space, then it will, just to make sure you take it, it will take the drop that is before, but the drop, that is after, but not the drop that is before. So you don't reach like this very big yield, but you are sure you fish all your cells, so you have a big, you have lots of cells of your interest, but mixed with others. Then you have purity, and purity is a lot more stringent. Basically, what the definition is that you should see only a cell here, and if you have a cell in this part of the next drop or the other one, it will just disregard this whole thing, just to make sure that you don't contaminate your sample. And the last one is even more stringent, is the phase. It's made for single cell cloning, and basically your cell has to fall into that measurement range, and if not, then it's discarded. So this is gives you very pure results, but the yield is really low, so you have to enrich dramatically your populations. Now if we put numbers together, that's an example. If you want to sort at high speed, and you have your target percentage of cells here, let's say you are interested in something rather rare, 1%, and you want, let's say, 100,000 cells, then you have to sort for 17 minutes. If you want to go even lower, then this time increases drastically. So for rare event cell sorting, you have to prefer initial enrichment method, because the time you spend here on the machine, your cells are sitting on ice and they wait to be sorted, and the one that got sorted, they wait that their brother and sister come and join them. So it's really bad for the quality of your afterwards studies. So you really have to minimize the time on cell sorter for having good quality. And here's just one example of the strategy we followed. We are part of the ImGen consortium. We sort for them CD4 T cells, naive T cells, and granulocytes. And here the key point is that from taking down the mice and collecting the organs, staining and all this, until cell sorting, purifying, we have to do that in 4 hours. So we have to be really fast. and optimize everything. And one of the key things, so the chance we had is that the population we look at, they are not so rare, so it's possible. But we do a first sort on yield, and we check very carefully the integrity of the sample, and we sort these. And then we do a second round of sorting, where now we sort on purity, and by doing so you will achieve 99 point, maybe you can't see it here. It's 99.7 at least % purity, so very top purity population. Now on your side, what is crucial is when you prepare your sample is the buffer in which you put your cell, because if the buffer is not appropriate, your cells start dying, or you put metabolic stress, and it's not good for what comes after. So you have these basic buffers, then we have other buffers that are meant for cells that are dying or where you have lots of dead cells. Then you have other buffers for adherent cells where you have you increase EDTA, for example, to avoid them to clump together. And sticky cells you can even put increase even more DTA and put FCS. Then the nozzles, 70 microns for high pressure cell sorting. So that's the more robust cell, primary cells. For example, then you have to shift and go to a 100 micron nozzle if you have transfected cells on another cell line, and you have to, and you can have medium pressure, and now you have to use an 100 micron for most of other cell line, big cell and things. Then you have to QC your data, of course. So this is something that should be done upfront of any kind of analysis you do. Kick out dead cells because you don't want to look at them. Kick out doublets. Doublets are coincident events that come together in front of the lasers. These are artifacts, so you want to kick them out. And then you can look morphological parameters and also look on time to check if the instrument is working fine for the measurement. Key thing is that if you put in garbage, you will have garbage out, right? So that's why all this is very crucial. And I think I am short on time, so any additional advice? Good sample preparation. Always use multiple doublet discrimination to really make sure you don't look at artifacts. Poorly separated population cause uncertainty in population discrimination, so are prone to making errors in judgment for the machine. You have to try to include both positive and negative selection criteria, and the more criteria you have, the better. So go high content multi-parameter flow cytometry. And again, very simple thing is never sort on an unstable string. And here is all the people we are working with, so people at CIF, people in the CIML, and people in the INPC, the Immuno-Phenotyping Consortium, the IMGen, and so. Thank you very much for your attention.
Thank you. One or two questions before the coffee break.
Most educational talk. I enjoyed it. It's always good to learn about the techniques we all rely on. Just a question about the multiple markers that you can use. You indicated you're up to 17 and perhaps up to 20. I guess that refers to colors. But is it really, at least for us that do transcriptomics, we do not want to fix the cells because we won't be able to measure the mRNA afterwards. How many markers do you think it's feasible to use if you're limited to extracellular markers?
I would say it depends on the instrument, because at the moment we cannot do more than 18 fluorescents. So the new instruments are coming up because you know what I told you at this cytometry conference. Someone say he made 26. So I mean the machine has to be in the pipe, and we'll relieve this. But for the moment, I would say 15 is achievable with work, but is achievable. And in many cases, people can do 13 without very big issues.
Yeah, but that's colors. But how is it really? Yeah, I was really. Can you really use 15 different antibodies without having too much cross-reactivity if you're stuck to using extracellular epitopes? Yeah,
you can do that. I mean, we do that every day, so it's working. And the other thing is that you can also kick out cells that are not of your interest, right? So that's one option. And also people are working, but this is even more complicated. if you define in your gating strategy, let's say your first 5 markers, they are super specific to your lineage, then you can use that as barcode for your staining mix. And you can have a whole hierarchy of other additional markers that will not be, maybe could be on others. But as your initial markers, they are in the one given cell line, cell subset, then you are specific. And like this, you can recycle all the colors. So that's the Sentinel approach. And you can reach 20, 30 markers at once. But that's complex.
I have a similar question going in the same direction. Is there anything known how much the high pressure is changing the transcriptome? And the second question is,
well,
many of the surface markers that you're using in the immune system transduce signals to the nucleus. How much is this affecting the transcriptome?
I have a very good answer for you. And I did some transcriptomic studies on T cell stages. And when I joined the CML first, we did not add a cooling system of the lines, right? And I was looking at T cell development at the really mature subset where you have commitment takes place between alpha, beta, and gamma, delta. So as a result of that, I had to stain for CD3. And CD3 is the main machinery driving the signal for the cell, right? And so I did my best of job. My staining was at 4 degrees, but then it was ultimately passing on the machine at room temperature. And what I could see on the fly is over the time of cell sorting is that my cells were getting activated and were joining the next stage of development. So I think if it's not cooled, then you have these very big issues. But if you keep it cool, not so much, because we then got this cooling system. And we did the cell sorting of our population, and we did the transcriptomic studies, and we had very clear signatures. Gamma deltas were gamma delta. The precursors that were not even expressing a gamma delta receptor were already expressing genes. So I'm highly confident that we had no issues due to this. Yeah. So cooling is the key. Yeah. And also don't sort for hours. Yeah.
There's a coffee break now, and then there will be the sorters afterwards. So like was said before, during the coffee break, please register for tonight.